and you're not going to really grade or do anything with them other than if you have questions or get something, we'll go over it. Otherwise, we'll just kind of, I'll go through this. Then I'm going to go basically give you some recap on the notes that we did once before. I put together another little presentation, and we'll get that too. All right, that said, let me find this. All right, let's see, first thing you're supposed to do is like 6, or 8 through 13, I'm sorry, on this one. So look at 8, it's test cross. All right, and the tutorial is really clear on this one. So basically what I want you to get out of the test cross is make sure you understand how to do this. This should be pretty straightforward for most of you. The idea is you have a dominant trait. Shut this. If you have a dominant trait like this is, whatever this dominant phenotype, two different genotypes possible, homozygous, heterozygous. So to find out what it is, you cross it with the homozygous recessive, little y, and that will give you either two outcomes. One, all are going to be dominant, in which case the genotype, one parent is homozygous dominant. If it's half and half, it's heterozygous, and that's the way you can tell. Really the only way you can do that is this way. That's the called the test cross, okay? Any questions on that? That should have been pretty straightforward. That's going to rehash it before. But actually, the next problem is the same thing. So if you do the first one, read the second one. Okay, the next one is the idea of one of the new terms that we talked about, incomplete dominance. And so this illustrates again using the snapdragons the concept that incomplete dominance is kind of a intermediate between the two dominant traits of the parents, or dominant recessive traits. In this case, it's red and white. In between is pink. And so the ones that show that are the guys in the middle, the heterozygotes. Remember that? And so these guys illustrate the phenotype that didn't exist before in the straight genetic dominance recessiveness, right? And so basically what you see is, again, is a third phenotype appearing. And basically what you'll see in the second one is totally something different than you expected before. You cross two of these again, and what you get is the next one, I think. Nope. Across two of these guys, and basically you're going to get a 1 to 2 to 1 ratio in the phenotypic ratio, and so that again illustrates incomplete dominance. So basically, the way you can tell incomplete dominance, if you can see them, as you see the colors different. If you look at the phenotype, it's going to show again a different heterozygous trait than before, because normally you've seen a 3 to 1, right? This is a 1 to 2 to 1, so it's different than the normal. Any questions on incomplete dominance? That's pretty straightforward. All right, the next one, the blood typing illustrates really two concepts. One is called codominance, and the other one is multiple alleles, and we didn't really talk about that yet. We'll introduce that today, but I'll touch on it right now. And, multiple alleles is a term in your textbooks, another one of those gene expressions out there. And what that means is that multiple alleles, you have more, it basically means what it says. You have more than 2 alleles for a given trait. So in this case, in blood typing, if you look, there's 4 alleles. So that's multiple alleles. Normally there's 2. There's either dominant or recessiveness and such. So this is this blood typing shows 2 different expressions of genetics. One, the codominance, and really you can't see the codominance until you get down to this guy, the AB blood typing. We have 1A and 1B. Actually, it's right here. That shows a codominant trait, in which case both parents express their genotype fully. And I'll show you that in a little more clearer way later. Okay, so in this case, so what you need to know is basically how to do blood typing. The idea here, this strange way that they express with the big eyes and the little eyes. The little eyes, remember, just means there's no allele at all. So if you have a dominant, or 2 dominant situations, you have big eye, or big B, big eye, or it can be heterozygous with little eye, big B, and a little eye. that's, basically that means it's heterozygous, okay? Yes?
Could you repeat how you would, if it said a woman with a type A blood and a man with a type B blood, how could you figure out if it was heterozygous or homozygous?
Okay, so this question right here?
Yeah, it says a woman with type A blood and a man with type B blood could potentially have offspring of which blood type.
Okay, sure.
I kind of just did it both ways and then just saw what was there.
So a woman with type A blood, the man with type B blood, okay? So the woman has type A
Yeah.
blood. So there's two ways of having A, right?
Yeah.
A, this his situation, or this situation. So
Yeah.
either of those would work for the female, the male. Can be, or. Okay?
Mhm.
So it says potentially have the following blood types. So basically, what are the possibilities here? All right? Well, if you do the Punnett squared,
Would we have to do the 16 square Punnett square?
no. So basically what you're doing is, what are the different possibilities of it having? Well, if you take a look at this, if you cross this guy with this guy, okay, and you basically have to do 4 Punnett squares here, basically.
Okay.
So you'd set up one with this cross, and
Yeah.
okay, in this case, they're all going to be AB blood, right?
Mm-hmm.
If you cross this one with this guy, okay, you're going to have the AB blood here. We'll just abbreviate this, and then the type A blood here. That makes sense. And then
Okay.
so you have 2 blood types. You got AB
An
and then
A.
A.
Uh-huh.
If you cross this
Then you're going to have
blood
B,
type, you're going to have AB again. If you cross this blood type,
A, B, and A.
can you see this okay from back
Yeah.
there, guys? I'm going to turn on the light. Okay, so this one's the IA here, IAB, so that's AB blood. Let me make this a little bit. You have IA, I, B, I, you're going to have I. So this one here is going to be type O blood, this one here is going to be type B blood, this one here is going to be AB blood, this one's going to be A. So really, this one's going to, you can get, if you have this cross and this cross, you can get any of the blood types.
Yeah.
A, and then the AB will be in this situation, right?
Okay.
So,
That's what I
depends
got,
on,
yeah.
on what the genotypes are as to what the possible phenotypes of the kids are going to be. Remembering again, if you have this one, you can do AB blood, A, B, or O. Okay? So all 4 blood types are possible from a heterozygous parent. If that makes sense. Did that
Yeah.
make sense to you?
Yeah, that's what I
Yes,
got.
ma'am.
Can you just do the bottom one,
Yeah, pretty much.
because that's what I do here.
Yeah, because that would include all the rest of them.
Yeah.
Yeah.
Look
Okay.
at that hot sweater.
Let's see, then we go into the next one. This is the one really that probably not gonna test you over. I just was throwing it out there,
Yeah.
showing you this is kind of a funky situation where in some cases you get, again, this bizarre scenario where the 2 alleles happen to be so caustic, they become lethal, and so they actually will kill the organism. And therefore, obviously, it kind of eliminates itself from the gene pool most times, right? All right, and back to, that was the first set. The second set was dihybrid cross. Basically, the same
You skipped
concepts were
13.
illustrated here, just using a dihybrid cross. The part that was probably pretty confusing was this thing with the lutein, and that was the epistasis question. These 2 questions both basically have to do the same. what it basically talks about is how one, this is probably one of the best places to show it, is how one allele, basically in this case the albino alleles, is little c's, they tend to mask the expression of other alleles. So when you see this, your ratio changes, your phenotypic ratio. Instead of seeing the normal distribution of like black, brown, into albino, you see a higher preponderance of this albino gene. And the reason is it affects the other expression in almost a dominant fashion, because it kind of mirrors, it hides this expression of this gene where it normally wouldn't. Okay. The next problem actually shows a little bit better, in my opinion. In this case, if you look at the ratio, normally in a dihybrid cross like this, your expected ratio, you guys should recognize this now, is a 9 to 3 to 3 to 1 ratio. But in the case of the epistasis, you see that basically that third one disappearing because the albino kind of dominates and is expressed in those three situations that would normally be brown over here. Okay? So in this situation, again, you basically the epistatic gene allele pair mirrors or masks the expression of the other ones, okay? And that's what you're going to see here. So what type of problem might you get? You might see something like this, and it'll say, how can you explain this variance from the normal situation? And you should be able to recognize that because there's additional genes that's different than incomplete dominance, right? So what you see here is this is called epistasis. And that's what I want you to understand. Okay? You won't have to do a problem like this. All right. Last thing we want to do is the sex-linked trait. And the classic examples that are used is hemophilia, and that's a real disease out there, and then color blindness. So you need to make sure you understand how to set one of these problems up, how to do it. In fact, we're going to do some pedigrees that are going to practice this a little bit more, starting in a couple of minutes here. And again, the idea is that you understand, start off and you do 6, right? 6 or 10. So the idea here again is that you have to know what male is, XY, female is XX. That should be pretty straightforward by now, right? And the idea that these traits exist on the X chromosome. So when you're dealing with the male, the Y really doesn't have any effect on the expression of that allele. So it only takes one recessive allele in males, if it's a recessive trait, to show out, versus the normal, which takes both. Remember that in previous genetics expression. So what you essentially see in sex linkage, you see a couple things different. One is you see a higher preponderance of recessive traits in males than you normally would. And secondly, you can have what's called a carrier. It's interesting, I have a new thing. That is, some females will carry the trait but not express it, and then they will pass it on to basically their sons at a 50-50 chance, depending on which chromosome your boys get. So that's kind of that's the big picture of sex-linked traits, okay? And then you got to do several problems, and we're going to practice a little bit more with that. Does anybody have any questions about sex linkage here? Is that pretty straightforward to you all? Yes, no, maybe so? Okay, again, this is pretty well done. The only thing that I think that they didn't do, I didn't like the way they did this, instead of putting the X's and the Y's in here, this would be like an XX, the XY. They put the little chromosomes, which obviously when we do our problems, we're not going to do that. We're actually going to put XX, XY. So that was the only really variance from this that you're going to see. That's where we practice on. Okay? All right. Well, if there are no problems, make sure, yes, ma'am.
Are you going to do any dihybrid crosses number 10?
So, let me get back.
Right there.
Sorry.
Do number 10.
Dihybrid cross, which one now?
Number 10.
Number 10.
I totally copied that answer. I just copied it.
Oh, link genes, yes. The thing about link genes, that was one that we mentioned a little bit, I think genes, instead of your normal distribution of independent assortment, you would see